Okay, good morning, students. Today we are going to talk about DNA markers, principle and applications. So this will put a total review on what are the markers available and how these markers have implication on present marker-assisted breeding or molecular breeding. So you already heard about a lot of marker system. So today's markers available all. Presently, the markers, DNA-based marker used in any molecular marker-assisted breeding or DNA fingerprinting are initially started with Sanger sequencing method. So originally, the Sanger sequencing been introduced to sequence each reaction can able to get a maximum of 750 base pair read length. If you do forward and reverse together and you align with maximum of, you can get 1.5 kb size, 1.5 1500 base pair size you can get. So these sequences been regularly updated in many genome databases like NCBI or EMBL or DDBJ or many international depository. The sequence are deposited. So the people start using the sequences. So the sequences being deposited are varying size from the earlier days, because earlier they used only a small set of sequences. Sometimes they sequence some express sequence tag ESTs or small clones, bacterial back libraries, or smaller size of clones which they identify for a specific research project on different crops. They try to submit in the NCBI ID. So by that way, the small amount of sequences are submitted, and those sequences being used for designing the primers and amplifying and try to use as a marker. So the DNA sequence output may be, it may produce like, it may be having the, either the sequence may be a small EST library or sometimes large scale libraries, yes, back libraries or EST library. So all the sequences are deposited in the gene bank and you can get a sequence ID. You already know that. The gene bank ID will be there. Every accessions you have smaller size, maybe 1 kb, 5 kb, 10 kb, is different size of DNA sequences available. So these sequences been submitted and it available in a FASTA format, and you can use these files for many purposes. For your mainly if you want to do design a primer for your specific marker, you can use it. So these sequences output, earlier days they started with smaller sequence output, and they try to deposit it, and the library is getting growing. So like that you will get EST library, back library sequences, EST sequences, and so many sequences are being accumulated and is available in the many databases. Later, when the sequencing is getting popular and many organisms they start sequencing, like the slides which I am showing here, every year one genome is sequenced. Like for example, in case of C. elegans or fruit fly or model crops like Arabidopsis, so every genome is getting sequenced every year. So by that, the amount of sequences accumulated is increasing in all the databases. So if you are, for example, if your Arabidopsis genome is sequenced, then the whole Arabidopsis genome sequence available. Similarly, for rice genome sequencing project completed during 2004-5, then whole rice genome sequences accumulated. So those sequences, if we have full sequences, so those full length sequences, you can use it for later now, the new generation sequencers, data is accumulated like so. So we can use it for SNP identification, etc. So every time when the sequences are accumulated, that been used for many times. Up to 2000, when you go earlier, the people used by back sequencing, that is clone by clone sequencing method and assembly. So they accumulated the sequence very less quantity. But after the when the NGS came into market and a new generation sequencer came into market, the people used the Sanger method and also they used the new generation sequencing technology, and sometimes they used hybrid technology. So this is by Sanger method. These are all the sequences being sequenced, by example, sorghum genome, soybean genome, 2012 barley genome, and also the, so these are all the barley, apple, potato, they used hybrid approach, both Sanger and also NGS. But in case of sorghum, soybean, and papaya, grape, these are all the genomes where people use the just Sanger method alone. Even peach, they used up to 2013. There are many genomes which been sequenced only by using Sanger, and after that hybrid method. And later, after 2011, this decade, 2011, mostly they use the NGS platform. So in that you can see watermelon, orange, chickpea, muskmelon, wild cherry, many genomes been getting sequenced. So now when you want to design when you want to use the markers for molecular breeding approaches for any breeding purpose. So now the people, these are all the crops which are recently sequenced by NGS method. People can easily access SNP data. So if you have the many, in individual crops, you have few varieties being sequenced, then it is easy that you can use those sequences and you can align the sequence and you can identify the SNPs and you can use it. So those crops and also other crops also, the sequence been aligned and the genome browsers available. This crop, there is resequencing also going on. For example, barley and wheat nowadays, they want to use more than 100 varieties. They are resequencing. Now, IPK Gattaslip and Germany, they use sequencing barley and wheat genomes, resequencing of varieties like rice varieties by very. So we can see resequencing will help you to find many SNP marker between the varieties. So this is why the people changing. So when the people, this is one of the famous crop genome circle. It been developed during Catherine Devos and Gail during the UK. They developed during 1998. At the time when they developed this crop genome circle, grass genomes, and they align all the grass genome, they used mainly RFLP markers with substitute with some of the SSR markers. So what they did, they made a consensus comparative map of 7 grass species. So all the 7 grass species, this is one of the classical work which been done during 1998. It paved the way for many of the comparative genome work and also synteny work, because they put a smaller genome, rice genome in the middle. This is the middle of the circle. When you see this, the rice genome been made into different blocks, each chromosome in individual blocks. That means rice chromosome 2 and 24, that is 12 linkage group, 12 pair of chromosome. So the 12 are inside the circle. The rice is inside the circle. Then followed by next circle, we have the foxtail millet. Then we have the red color is rice, foxtail millet. Then we have sorghum, we have maize, and we have sugarcane, and we have oats and triticale. So every circle represents different. So for example, the rice chromosome 1 and the barley chromosome 3, they have this synteny. The rice chromosome 1 and barley chromosome 3, they have synteny. Similarly, every chromosome, they have the commonality between cereals. So this picture shows how the cereal genome, all the 7 grass species, when they are aligned together in a circle, how they share the genome sequences between the grass species. How many chromosomes they have macrosynteny, how many regions there is a microsynteny. So all the things that people can able to understand how the cereal genomes are have the highly synteny nature. So what this information will help us to derive some of the markers for our molecular breeding. So when you see a evolution point of view, when cereals are separated into maize, all cereal species like maize, rice, wheat, barley, and foxtail millet, which are all evolved from a common ancestor. So the common ancestor is one, from that they are branching out. During the course of evolution, when the cereals been branched out, they differentiated into different species. That means initial period, all are originated from common ancestor. So that's why they share maximum homology. So when you develop a marker, when the rice genome is sequenced, first rice genome is sequenced during 2004, and the rice genome sequence will help us for other genomes during 2004-5, other except rice, no other genomes are sequenced. So this genome sequence help us to develop markers for other species. So for example, SSR marker, when you develop the SSR markers of rice, if you take it and use it for other grass species, its cross transferability of marker to other species is, you can have 10 to 20%, you can transfer the markers from one species to other species. That means the repeat regions which are present in rice genome, which is common, which is present in other genomes also. Similarly, they try to use the genomic information and try to enrich the information in other species. So that's why this crop genome circle help us to know which are all the, now you have almost, during 2020, you can see almost sorghum genome sequence, foxtail millet genome sequence, maize is sequenced, triticale sequence. So all other genomes are sequenced. Now also you can align the sequences. You can able to know that how many sequences are homology and which are all the regions you can work on that. So this way this help us to, common centenary region help us to design the markers. That's why now the SNP discovery is easy when compared to 10 years or 20 years back, because every genome is aligned. Each genome, there is a resequencing of more than 50 or 100 varieties been resequenced. So if, for example, rice genome Nipambare sequence available, then you have other 6 individual varieties being sequenced. A particular region, for example, you have 400 to 700 base pair, a small region. If you take any chromosomal region, you can see 1, 2. Every 100 base pair, you can have at least 1 SNP. So every single nucleotide changes, you can observe in all the species. So this help us, the resequencing will help us to find out this a single nucleotide variation among the varieties. So you can design the assay based on that. You can genotype it, fingerprint it, or mark rested breeding, you can use it. So that's why people use the SNP discovery program. Okay. Coming to the markers, which when you are now at the era of sequencing era, and you have very huge information on the sequence data and SNP data available, but when you look back into the previous history, at least you should know what happened in the previous days. First, they started with RFLP markers, restriction, fragment, length, polymorphism. You already heard about that. So RFLP marker is the first marker being used, because those days we don't have the sequence information on the crops. So there only you know the DNA and you have some restriction enzyme. So the people use this RFLP technology by using genomic DNA, restricted with some of the enzymes, and they developed a fragment length polymorphism. They used RFLP. After that, once the PCR machine been invented during 80s by Carey Mullis, then the amplification-based marker came. That is, the first one is RAPD, random amplified polymorphic DNA, then followed by SSR, simple sequence repeat, then ISSR, inter-simple sequence repeat, then SCOR, sequence characterized amplified region SCOR. So these are all the amplification-based marker which ruled 2 decades. These markers are used very commonly, but nowadays very limited RAPD some cases, and also SCOR markers, some specific purpose people are using. RFLP is almost vanished from the scene because of some of the difficulties. Then the amplification-based marker came, then they combined with the RFLP, they developed a new marker, the AFLP marker, that is amplified fragment length polymorphism. It combined the restriction-based marker advantage and also amplification-based marker advantage. They combined together, the AFLP marker came. So AFLP marker we ruled around another 2 decades. Then after that, new generation marker came, that you know that a single nucleotide polymorphism marker, which is very commonly used nowadays. So, and also you have to see when these changes happens. When you read the literature, when you look on the papers, when you refer for a particular markers usage, you can see that during 1900, 1990, up to 1995, 2000 or 2003. For example, one period during 95, when the 95 it started, microsatellite marker started during 90 itself. Then SSR marker still it's have simple sequence repeat marker still have major share from 1990 onwards. It started initially because sequence, many of the sequences are start accumulating from 90s. and whenever the sequence are getting accumulated in the databases, then the availability of SSR, finding the possibilities of SSR repeats are high. So that's why the people start using the more SSR marker in many species, many crops they use. So now it is a major share at microsatellite marker. So when you come to 1995, so what are all the markers which the share is reduced? That is mini satellites, RFLP totally getting reduced. So up to 2000, it is totally vanished. Very few applications. Then if you see that SNPs is getting bigger. So the SNP share now after 2000, it completely expanded. Now 2020 is the biggest marker system. RAPD is have very little usage standard. It have some kind of usage, still it is going on, because it's there is the species which have no information, no markers available. The first attempt is to go for RAPD markers. So this is why the people using that. So these are the markers. I can say that how the relative importance of markers during the period, every decade, every 10 years or 5 years, if we take this case, every 5 years, check that the pattern of shift in the paradigm shift in the markers usage. So now only the maximum usage of SSR marker and the SNP markers and little usage of RAPD markers. Okay, so just a few discussion on the markers which is already the used markers. First one, I can say that how the RFLP markers, just you know the principle, it have usage in the later stages also. So already many of you learned about the RFLP markers. It's a restriction fragment length polymorphism. I will tell you the major principle on that. If you are interested, you can go deep and work on that. When the RFLP markers in the market, the question is only you have the DNA available, so you have to isolate the DNA. Those DNA been restricted with restriction enzyme. They used whatever the available restrictions enzyme they used, like normally equal around MSC1. They use restrict. When you restrict the genomic DNA, you can run in agarose gel, you can get it a smear. You cannot see the discrete band because when you digest it and run it, it's very small quantities there. You cannot amplify those days. The DNA cannot be amplified. You don't have that PCR machine. So when you restrict the DNA, you look like a smear. So what they did, the restriction fragments been transferred to the nylon membrane. That is we call southern transfer. Southern hybridization technique is very common those days. Though the transfer of the cold DNA, the restriction fragment into the nylon membrane, they put a wad of paper in the top. Bottom, we have the gel support, and then the buffer will be there bottom. So by way of capillary movement, the DNA available in the gel is transferred to the nylon membrane. Once it's transferred to the nylon membrane, then these fragments available in the nylon membrane, you cannot visualize. So for that, they go for probes. So you have to hybridize with the probe, that radioactive label probe. Then if you hybridize it, you can visualize by autoradiogram. So the probe is one, the known DNA fragments. The probe preparation itself separate step. Any DNA, genomic DNA digested, and these fragments are separated based on size. Then these target fragments are inserted into vectors. Then they have the recombinant vectors cloned in the bacterial system. Then these clones are multiplied, and these clones are multiplied and served. So this is a known fragment which we served as a vector. Then if you these clones, when used as a probe, you have to treat with radioactive label these fragments. And then if these probes are hybridized with your nylon membrane, then those hybridized fragments, you can get the signal. Because it's already labeled with P32, or this is radioactive label. So those hybridized only, you can get the signal in when you expose to X-ray. So this kind of X-ray film, you can see the dots. You can here also again, this is a codominant marker. For example, if you have one parent, if you have the two different parents, you have some parents, you have for top allele one parent, bottom allele another parent. Hybrids, you have both alleles. So this is a codominant marker. The people used this one for the differentiation of varieties or construction of linkage map that people used. So here we used specific principle when the DNA, when you restrict with the restriction enzyme, if we do two varieties, for example, this particular variety and second variety, this restriction enzyme, they made a three cut in using this particular variety, but another variety make only 2 nicks. So this DNA, they have 3 nicks, this have 2 nicks. So because of that, this generate bigger size fragment, this generates smaller size fragment. So when the probes are hybridized, you can see this have bigger size fragment, it has smaller size fragment. So you can easily separate it, and by that you can differentiate the variety 1 and variety 2. Like that you can use it for fingerprinting purpose. So this way people used RFLP markers for the earlier days. They used only, because we don't have the PCR based markers in the market, so that people used only the RFLP markers. The second thing is, people start using, when the PCR machine came, they start using the PCR based marker, because RFLP takes time, because it takes time, at least 1 week time, starting from DNA extraction. Then you have to second stage, you have to go for restriction digestion, then you have to southern transfer, then you have to go hybridization, then autoradiogram, finally you get the results. If you start the experiment on Monday, you will get the results on Saturday. So now if you go the PCR based marker, within the 3 hours you are getting the results. So that's why the people start shifting to the PCR based markers. So coming to the PCR based markers, so you have PCR based marker. The first marker came into picture is RAPD marker, random amplified polymorphic DNA. So here, once the PCR been invented, people take the DNA and they design. Again here, RAPD marker, there is no need of sequence information on a particular crop. So that time there is no sequence available. During 80s, many crops we don't have the sequence information. Even rice sequencing is completed 2004. During for example, during 86 or 90, when Williams 90 he used many RAPD markers for the fingerprinting work. Those time there is not much sequencing on any crops. So what they mainly for RAPD markers, they mainly depend on, you take any random sequences, synthesize a primer. This case 10 mer or 15 mer, you design a primer. It is only random nucleotides. So the primer will be random. So that's why they put Opron primers. They made order the random primers, again make some arrangements. They make Opron primers. They started using that. So when you use this random primer, when the primer amplify wherever in the genomic region, if you find the complementary sequence, it's bind there, it will amplify. It's only one sequence. There is no forward and reverse primer. So wherever this RAPD primer bind on the complementary sequence, it's try to start amplifying. So by that you will get a lot of multiple alleles you will get. So same sequence may be present in 10, 20 position in the genome. So they get amplified. So you can get a lot of fragments, maybe 10 or 15. Any RAPD gel, if you see more than 10, 15 fragments, you can see. Among the all fragments you see, as for example, this case, the bottom two are monomorphic because all have this particular fragment. But this fragment present in A, absent in B, but heterozygote is present. So this is kind of dominant markers. You can differentiate. You cannot differentiate the homozygous dominant and heterozygous one. But this case, the presence of this particular band presence, absent present. So this is a polymorphic. So you can use this as for a fingerprinting purpose or any other purposes. So this is the one typical example. We recently, our undergraduate student worked on fingerprinting of lucerne and thinai. So even though the lucerne is a very fodder crop and is less genomic information available, we tried to use the RAPD markers. So these RAPD markers can differentiate the COFOR variety from Anand and other varieties just by differentiating one allele. There are many alleles amplified, but there are few alleles, only 1 or 2 fragments which can differentiate. This case also DNA fingerprinting of thinai varieties. There you can see the COTE7, the released variety, and the newly proposed culture. There is a difference in the RAPD profile. So this way the sequence informations are not available. So they try to use these RAPD markers for the fingerprinting purpose, or earlier they used for mapping also. So now thinai some information coming for sequencing already completed. So you have a lot of SSR markers available. So if there is no markers available with you, just you can try with the RAPD markers. we can get polymorphism. Only problem is RAPD. If we are not using stringent amplification condition, that according to, if you have proper annealing temperature and the change in the some kind of annealing temperature or other thing, PCR conditions, the bands looks different. One pattern cannot be repeatable. So that's why people not using RAPD markers regularly. So with this, the RAPD, RFLP, and the kind of markers which we are using have a lot of implication on the DNA fingerprinting purpose. Okay, we will, with this, I will stop this presentation. We'll start a new sessions shortly. Thank you.